Hello everyone, and thank you for joining this virtual session. I am presenting the research project titled Polymorphism Analysis of GDF9 and BMP15 genes and their association with litter size in Kajang Bor Crossbridge Ghost in Sabah, Malaysia. This project was conducted under the UMS GREAT grant at Faculty of Sustainable Agriculture, University Malaysia Sabah. As a quick background and significance of this study, goats are a key component of Malaysia's livestock sector, contributing on food security, rural income, and low methane emissions. However, this sector highly affected with many factors, among them genetic factor, husbandry practice, and environmental factors are highly affecting the productivity of goat production industry. So enhancing reproductive traits such as litter size is vital for improving productivity and sustainability. So for this study, we focused on two fertility related genes such as GDF9 genes and BMP15 genes, which highly play crucial role in ovarian function and follicular development. For this study, we had two objectives. The first objective was to identify polymorphism of BM15 and GDF9 genes in Kajang Bor Crossbridge goats in Malaysia. The second objective was to assess the genetic diversity and its association with litter size in Kajang Bor Crossbridge goats in Malaysia. For this study, we followed a number of methodologies. The first methodology was to collect background history of the female goats, such as litter size and parity, and collecting blood sample from those animals, and the 5 ml blood samples were collected from each animal, and we extracted DNA from those animals, and we conducted PCR RFLB followed by a Sanger sequence at Apical Scientific Malaysia, and we conducted association analysis to associate the phenotype trait with litter size. For the genetic diversity, sequences were aligned with the Caprin GDF9 and BMP15 using the reference ID at the INCBI, and statistical analysis were conducted to evaluate genotype frequencies, allele diversity, and association with litter size. From this project, we identified 4 novel SNPs, which are G320C, A352G, G366T, and G375T. SNPs were identified in exon 2 of GDF9. However, no SNPs were identified in exon 2 of BMP15 gene. The AG genotype at A352G and GG genotype at G375T were significantly associated with higher litter size. And all identified SNPs lead to amino acid changes, suggesting functional impact on ovarian development and fertility. The genetic diversity parameters such as heterozygosity, effective allele number, and polymorphic information content were calculated, and the Hardy-Weinberg equilibrium tests showed no significant deviation from the studied population of this crossbreed goat. These markers could be used as a marker assisted selection to improve reproductive performance and economic returns in this crossbreed goat farming. Therefore, this study will have an impact and implication. Therefore, AG and G genotypes can be used to optimize the breeding strategies. This contributes to livestock self-sufficiency of the country, rural income growth, and sustainable agriculture in Malaysia. The findings support genetic improvement programs aimed at enhancing meat and milk supply by integrating molecular tools to breeding. It can accelerate genetic gain and improve goat productivity. In conclusion, GDF9 polymorphisms, particularly the two markers, which are AS352G and G375T, show promise as a genetic marker for improved litter size in these crossbreed goats. This research lays the groundwork for marker-assisted selective breeding and genomic selection for better breeding gain in Malaysian goat breeding industry. Therefore, however, further investigation to other candidate genes such as Eas 15 and the rest fertility related genes should be conducted to assess the role on fertility and litter size on this crossbreed goat. Thank you. We thank UMS for funding this project. Thank you for your attention, and I welcome any questions or feedbacks. Thank you again.